evolution say people came from monkeys. And the question is, why is there still monkeys, you dumb motherfucker, you? Is these the retarded monkeys? They didn't turn into people just yet.
No matter how life began, on the backs of crystals or in the test tube of some intelligent designer, everyone agrees it started with a single cell. But what is a cell? Let me ask you a question.
Yeah.
Darwin wrote The Origin of Species in 1859, published it in 1859. He had an idea of the cell as being quite simple, correct?
Yeah, everybody did.
Okay. If he thought of the cell as being a Buick, what is the cell now in terms of its complexity by comparison?
A galaxy.
If Darwin thought a cell was, say, a mud hut, what do we now know that a cell is?
More complicated than a Saturn V.
So what is in a cell as far as we know now?
A world that Darwin never could have imagined.
I needed someone who could give me a glimpse into this world. So we went to molecular biologist Doug Axe.
Think of a cell as being a nanofactory, a factory where, on a very small scale, digital instructions are being used to make the components of the factory. Here we have the famous DNA double helix. You can see the two helical strands that are intertwined and wind around each other on the outside of the molecule. This is the material that stores all of our genetic information. In higher life forms, this will be the equivalent of something like a gigabyte of information stored in the molecules that form the individual chromosomes, all packed within the nucleus, which is a tiny fraction of the entire cell size. So what does DNA do? Well, the information in DNA ends up providing the information for sequencing the amino acids to make proteins. We have information in a one-dimensional form that provides the information for a three-dimensional form.
Welcome to the Casino of Life. Who wants to spin for a chance to win?
Oh, sure. I'll give it a shot. What do I win?
Take a look at this.
Huh? How about the world's first single-cell organism? This perfectly aligned string of proteins could be yours.
Now, take a spin.
I
won! Tina, tell him how many times he needs to do that to win the prize.
250.
That's right, folks, and all in the correct order.
But that's impossible.
We've heard that before, haven't we, Richard?
Come on, Mother Nature, do your thing. You stupid machine! I hate you!
So, in the game of life, it looks as if the house always wins.
We're talking about something that's staggeringly improbable, roughly 1 in a trillion, 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 trillion.
Let all of life choose a million, a trillion, trillion. The number is essentially zero. Something has to skew nature to choose the ones that work.
The language of DNA is digital, but it is not binary code like a computer, where binary encoding has zeros and ones to work with. DNA has 4 positions, T, C, G, and A. These letters are used to represent the 4 informational nucleotide bases of a DNA strand. Whereas a digital byte is almost 8 binary digits, a DNA byte, called a codon, has 3 digits. Because each digit can have 4 values instead of 2, a DNA codon has 64 possible values, compared to a binary byte, which has 256. When you take into account that all 20+ million species of known life and each of their subsystems and sub-subsystems are all produced within this 64-value coding language, you can see the amazing efficiency level at which the genome works. Of the 20,000 to 30,000 genes now thought to make up the human genome, most cells actually express only a very small part of the total information code, which makes sense. A liver cell, for example, would have no need for the DNA code that makes up neurons. But since almost all the cells carry around a full copy of the genome, a system is needed to define out information that is not needed. And that is just how DNA works. Just like a computer program, the genetic code is full of if and end if statements. This is why stem cells are so hot right now. These cells have the ability to differentiate into everything. The code hasn't been defined out yet, so to speak. Stated more precisely, stem cells do not have everything turned on. Over the lifetime of the stem cell, during which time it may clone or fork many times, it specializes itself. Each specialization can be regarded as choosing a branch in a tree, in which it will ultimately and finally operate and specialize. Each stem cell can make or be induced to make decisions about its future. Each decision makes it more specialized. DNA is not simply a computer programming language, but as you have already seen, there are some startling analogies between DNA and computer code. We can view each cell as a CPU, or a central processing unit, running its own kernel. As you probably know, in computing, the kernel is the main component of most computer operating systems. So each cell has a copy of the entire kernel. But amazingly, each cell chooses to activate only the relevant parts, which modules or which driver it loads, so to speak. If a cell needs to do something or call a function, it calls up the right piece of the genome and transcribes it into RNA. The RNA is then translated into a sequence of amino acids, which together make up a protein for which the DNA coded. Now for the truly amazing part of the equation. This called up and coded protein is then tagged with a shipping address. This address is a marker consisting of several amino acids which tells the rest of the cell where this particular protein needs to be deployed. Once the protein arrives at its intended location, the delivery instruction is then stripped off and several post-processing steps are performed, activating the protein to now do its job. The protein is not activated until it arrives at its marked and intended location. The amazing and complex intelligent communication functions of DNA are myriad. They are too numerous to recount and explain in a short and simple video such as this. But the fact remains, purposed and directed information exchange is continuously conducted within the human genome and the DNA-RNA functions that make it up. Purposed and directed information exchange.
I'm finally just beginning to grasp the complexity of the cell. Are there systems within the cell that go well beyond Darwinian evolution? Some type of cellular technology that drives adaptation, replication, quality control, and repair? What if these new mechanisms have massive design implications? Well, I say, so be it.
Did you know that in the cerebral cortex alone, there are roughly 125 trillion synapses, which equals the number of stars in 1,500 Milky Way galaxies? The human brain has more switches than all the computers, routers, and internet connections on Earth. Does this sound like the results of millions of years of evolution, or the work of an intelligent creator? But no matter how far up you go in the evolutionary tree, there's still the question of how that first whatever it is came to be. This entire theory of evolution and the last universal common ancestor hinges on the assertion that non-life can create life. That is, chemical elements are able to spontaneously combine to form life itself. Non-life becomes life. Is that even possible? Can life arise from non-life? Even though scientific experiments have so far been unable to create life, most evolutionary models require that spontaneous generation of life happened at least once at the very beginning. How likely is that? The famed British astronomer and avowed atheist Fred Hoyle calculated that the chance of obtaining all the enzymes required for life at random is outrageously small. Comparing it to the chance that a tornado sweeping through a junkyard might assemble a Boeing 747 from the materials therein. Hoyle and his colleague then offered up alternative hypotheses that life began in space, transported to Earth by comets, meteorites, and other cosmic debris.
Effectively, what they're doing is saying, we don't have the answer. We can't figure out the conditions to evolve life on this planet. And really, it's impossible. Laws of chemistry and physics forbid it. And so if we can push it off to the unknown, where we don't necessarily know the chemicals present, the interactions at play, the physics at play, then maybe there's a better chance. And all we need then is an accident to bring it here, and we solve the problem. But all that does is push the problem one step backwards, and we're back to square one, which is, well, how did life evolve on that planet? Presumably the laws of chemistry and physics are the same wherever you go in the universe. So it doesn't really solve the problem, it just avoids it by pushing it off to somewhere else.
Some of the basic elements that are required for life are things like carbon, nitrogen, phosphorus, sulfur, oxygen, and that's just to name a few things. And obviously, all of those things are important components of what we call macromolecules, which are a very important part of every living thing. And for example, we have nucleic acids, which are obviously parts of DNA and RNA. And DNA is really important because it contains the information for all of the life processes that go on in the cell, and it's necessary to be replicated in order for the cell to divide and pass on that genetic material. And then we have things like proteins, and they're really the workhorses of the cell. They're what does the job, so to speak. They allow metabolism to happen so that the cell can break nutrients down and build new things up from that. And then, of course, you have carbohydrates, which are things like sugars, and they really provide the energy for the cell. So those are just some of the basic things that are required for life to be able to exist and to continue.
DNA, RNA, and proteins are biomolecules, which together are essential for life. DNA and RNA are needed to make protein. Protein and RNA are needed to make more DNA. And DNA and protein are needed to make RNA. So, which came first? So those are some of the biomolecules that define living things. In the absence of any one of these, life does not exist. But how did these biomolecules supposedly randomly combine in the right environment to start life on Earth?
When we look at humans, it's really interesting because even the simplest things are extremely complex. And one of the examples of that is the blood clotting pathway. Obviously, that's very important to us. So when we get a cut, the blood clots and we don't bleed to death. And when you look at how that happens, it's a chain reaction, so to speak, of many, many different proteins, or what are called factors, that are involved, that the end result, one kind of activates the next one, which activates the next one, and the end result is a blood clot. So by definition, the blood clotting system, or the blood clotting pathway, is irreducibly complex because it's made up of many different parts. And if even one part is missing, if even one factor is missing, blood doesn't clot. Those types of systems, those types of complex biological systems, or irreducibly complex systems, are very hard for evolution to understand how those things came about, because all they're really left with is mutation and natural selection. Those are the main mechanisms for Darwinian evolution. And natural selection, well, it only selects from what's there. It can't create anything either. It can't account for the origin of novel things or new things. It can only select or cull, so to speak, from the proteins or DNA or whatever it may be that's already there.
You decide. Random, traumatically mutated, non-purposed, accidental evolution, or intelligent design? Any truly intelligent person knows the intelligent answer to that question.